everyone for joining us this morning. I have the pleasure of introducing Dr. Wolozin, who is a professor of pharmacology and neurology at Boston University School of Medicine. Dr. Wolozin completed his undergraduate degree at Wesleyan and earned his MD PhD from Albert Einstein College of Medicine. He did postdoctoral fellowships at Mount Sinai and NIMH before heading to Loyola University and then eventually BU in 2004. He has received numerous awards, including election as a fellow of the AAS and Spivak Distinguished Scholar Neuroscience Award at BU and the Zenith Award. The Wolozin lab studies the dynamics of RNA binding protein aggregation and the contribution dysfunction of RNA granules makes to neurodegenerative disease, and uses a variety of models to study the disease, everything from primary neurons and ipsies to C. elegans and transgenic mice and human tissues. I am super excited to see him for this talk as a avid reader of Dr. Wolozin's papers. It seems to pop up all throughout my research career, so I am incredibly pleased to welcome him to speak.
Well, Isabel, thank you for offering me the invitation. I wish it would have been great to actually come to UCSD and see everyone and everything, and maybe, as Eddie said, in future years we'll be able to do that. I'm going to talk today about membraneless organelles, RNA granules, and RNA metabolism, but in the context of tau and Alzheimer's disease. First, I have to share with you that I have a conflict of interest because I'm co-founder and chief scientific officer of Aquina Pharmaceuticals, which is a company that's designing drugs to treat AD and ALS. So on with the show. So when we think about dementia, it's a general behavioral kind of phenomenon that we describe, but the contributions are multifold from many different. We talk about Alzheimer's disease, but in addition, vascular components contribute a lot to it. UCSD is like a huge ALS group, so ALS can contribute to dementia, frontotemporal lobar dementia, and of course, patients with Parkinson's disease or develop dementia. So in order to understand the various contributions of this, one looks at the kind of major pathologies that accumulate. I'm going to be focusing on tau. And so we're all on the same place. I know that most of you know a lot about Alzheimer's disease, but just to give you a sense of the most basics. So amyloid precursor protein produces beta amyloid, which accumulates outside the cell as oligomers. Those oligomers fibrilize, and in the process appear to stimulate an inflammatory response that gives rise to reactive microglia and eventually to these neuritic plaques. The combination of the inflammatory response and the A-beta oligomers act on the neuron to stimulate toxicity and injury. And in Alzheimer's disease, that injury appears to be manifest and proceeds through tau, which oligomerizes and then fibrilizes to form tangles, gets phosphorylated in the process. So when we've thought about the disease, it's kind of just accepted that tau develops pathology, phosphorylates, oligomerizes, forms tangles. But you don't hear much discussion about why that phosphorylation and the subsequent accumulation of somatodendritic compartment how it might contribute to the cell. Is this a mistake or is this part of a biological process? So we've been investigating this. The first place to look are the genetics of Alzheimer's disease. And they really don't tell you much about tau because the genes that are the most highly penetrant all seem to affect A-beta production. And then you have some inflammatory genes, TREM2, and some cholesterol-related genes. And the genes, when you get to tau, you see only very modest effects of tau and tau-related genes. So that says to us that in Alzheimer's disease, at least from a genetic standpoint, we need to consider A beta, inflammation, cholesterol. So where does tau come in? The beginnings of an insight into this, at least for me, came from looking at the ALS frontotemporal dementia spectrum. So when we look at ALS and frontotemporal dementia, what we see is the genes that cause disease fall into a number of categories. One important category are RNA binding proteins. And then there are genes that relate to proteostasis, and then there are inflammatory genes. But when we look at this, we also see tau. And the question is, what is tau actually doing there? So, looking at the tau in the realm of RNA binding proteins begins to generate some hypotheses. RNA binding proteins are really interesting class of proteins because they form complexes through a process called liquid-liquid phase separation, and they form what are called membraneless granules. And many of you have seen these membraneless granules in the cell. If you've looked at a nucleus and seen the nucleolus, have you ever stopped to pause and think, why is that nucleolus round? Why doesn't it just dissociate? And the answer is it's a liquid droplet within the nucleus. And all the RNA there, combined with the RNA binding proteins in the nucleolus, help to promote this, to coalesce to form a membraneless droplet. In fact, virtually all the complexes in the nucleus are liquid-liquid phase separated membraneless granules. And throughout the cell, wherever we find RNA binding proteins, we find many membraneless granules. But the ones that seem most relevant to disease are these granules called stress granules, which only accumulate in response to stress and contain many nuclear RNA binding proteins that translocate out to the stress granule in the process of their formation. And many of these proteins are actually proteins that have genetic links, usually to ALS or other motor neuron diseases, as shown here in orange. Study of the biophysics of membraneless granules and liquid phase separation begins to provide insight into many, many aspects of cell biology. First of all, on a more general level, the fact that these associate through phase separation answers a fundamental question about the cell, which I don't know if you have ever wondered this, but I think of the up to 1 million proteins in the cell when you consider isoforms. It seems so complicated. How do things actually find each other? It seems crazy. We know it works because we're alive, but it seems crazy. One of the answers is that like associate with like in liquid droplets, which are these protein phase separated protein droplets. How do these form? Well, RNA binding proteins combine with RNA and kind of weakly associate. I'll tell you in a moment how that happens. And this gives rise to this phenomenon. You can actually see just with a microscope with recombinant proteins where you see these protein droplets that form. But this system has an Achilles heel because the protein droplets occasionally go through conformational changes to form much more stable complexes that are solid. And these solid aggregates begin to accumulate. And there is some evidence that this gives rise to some of the pathologies that we see in neurodegenerative diseases. So this process has an Achilles heel. When you're young and you have a strong catabolic autolysosomal system, you can get rid of this stuff. But as we get older, our ability to catabolize these aggregates decreases, and so the risk of having them accumulate increases. The biology of phase-separated protein droplets is really quite simple. You basically can think of it as stickers and linkers. And so these low complexity domains have these portions that kind of weakly associate, and they have linkers in between that kind of connect them. The longer the linker, the less the association. The more stickers, the more the association. And so you have multiple weak links that are bringing things together so that they're always touching each other and associated, but they're very dynamic at the same time, hence the term liquid droplet. And you can draw free energy curves to show kind of the tendency of these to form, and they really follow these curves quite well. So this is an example of a biological phenomenon that can be almost completely explained from first principles by physics. So these RNA binding proteins form these stress granules. Early on, we went and looked at markers in frontotemporal dementia and Alzheimer's disease to ask the question whether tau might also associate with these stress granules, because we saw that tau kind of was in there with other RNA binding proteins as a cause of disease. So right away, we observed that tau strongly colocalizes with TO1 and some other RNA binding proteins, but not all RNA binding proteins. For instance, G3BP doesn't colocalize well with tau. In fact, at the ADP meeting in Barcelona this year, Ravi Georgievich gave a great poster where he showed that in fact G3BP associates with non-phosphorylated tau, but doesn't associate with phosphorylated tau. So it begins to explain part of that biology. We followed this up and showed in vivo that if you reduce TO1, you can actually impact on the pathophysiology of tau in a transgenic mouse. When we reduced TO1, we showed that the amount of, this is a mouse that has P301S tau, so it accumulates tangles. And with that, you get death of hippocampal neurons as well as other neurons. And what you can see is that if you, in a normal mouse, you can see this loss of these hippocampal neurons by 9 months. But if you reduce TO1 by half, you kind of can preserve the amount of neurons as quantified here with 2 independent methods. So we're wondering why that was happening. And we did biochemical studies and found, to our surprise, that one of the most striking things was that actually, if you reduced TO1, you actually accumulated more fibrillar tau, which seemed contradictory until we looked at oligomeric tau. And what we saw was that a normal P301S tau mouse has a lot of oligomeric tau, while the P301S animal crossed to a heterozygous TO1 mouse has very little oligomeric tau, but a lot of fibrillar tau. So this suggested to us that perhaps the amount of oligomeric tau was actually what was being modulated by TO1, and that we could, and that it was changing that ratio of oligomeric to fibrillar, and that was leading to some of the protection. We tested this in vivo by taking a P301S mouse and injecting either an oligomeric tau fraction, as shown by the S1P fraction taken from a P301S brain, or a fibrillar tau fraction. And we injected them into the CA1 and looked at it both in the ipsilateral CA3 and in the contralateral entorhinal cortex. What we saw was what we expected. You know, the field kind of knows somewhat that oligomeric tau is more toxic, and in fact, that's what we saw. If you look in the lateral entorhinal cortex after 3 months, there was a reduced amount of neurons in brains injected with oligomeric tau, whereas the fibrillar tau brains had pathology, as I'll show you, but didn't have much degeneration, and this is quantified here. When we crossed this, looked at the animals crossed with the heterozygous TO1, what we saw was that that in a normal mouse, P30 mouse, we could see a lot of oligomeric tau, as shown by Kayed-Marquez's Marquez-Kayed's TOMA2 antibody. But when we looked in the animals where we had reduced TIA1, there was very little TOMA2, suggesting that indeed the reducing TIA1 was getting rid of the oligomeric tau. And when we looked at where the oligomeric tau was showing up in the contralateral cortex, what we saw was that the oligomeric tau associated with other stress granule markers such as TIA1, EIF3 eta, PABP. But interestingly, the P3 fibrillar tau didn't associate with these RNA binding proteins. And in fact, the fibrillar tau accumulated in a different localization in the cell, more out towards the synapses. So this kind of really emphasized the strong connection between tau and RNA binding proteins. Sonia Han and Ken Kosik's group showed that, like RNA binding proteins, tau also undergoes phase separation. And this has now been reproduced by many groups, including Suzanne Wegman's group, Marcus Switzer, Schweckstetter, as well as our group. So this suggests that tau falls into this kind of category of proteins that readily undergo phase separation. But phase separation, as done by these other groups, is kind of involves putting recombinant tau into a dish, adding some polyethylene glycol or phycol, which is not biological, using high concentrations of everything, and seeing that it forms droplets. And that to us, at least to me, never seemed, it was interesting, but it wasn't biological. So we went, and Peter Ash in my lab went and said, well, what if we start combining tau with RNA binding proteins and RNA? What happens there? Well, in that situation, what we showed was that, in fact, tau is miscible with the TIA1 droplets, and actually at higher concentrations forms subdomains within those droplets. What's also interesting in this same study is that we compared it to other proteins like G3BP and showed that tau actually preferentially is miscible in TIA1 as compared to other RNA binding proteins, which begins to provide some insight into why that tau TIA1 stress granule connection. And then using super resolution imaging, we showed that if you have a low amount of tau, it's kind of generally miscible, but with increasing concentrations or increasing time, it vitrifies and actually pushes the TIA1 away and kind of changes conformation. Now, the result of all of this is that it turns out we showed that TIA1, in fact, inhibits fibrillization. This we could show in vitro, where if we combine TIA1 with tau and RNA, if you don't have TIA1 there, you get lots of fibrillization, but if you do have TIA1, you don't get fibrillization. We showed this by EM, and when we quantified the amount of oligomer by ELISA, we could clearly show that TIA1 stabilizes oligomers and promotes their accumulation. So this begins to kind of draw attention to the strong connection between RNA binding protein stress granules and tau oligomerization, which we've now shown is more toxic than the fibrils, which are the classic pathology. So we wanted to understand, well, okay, if it's oligomers we're talking about, what is the biology of oligomers? To explore the biology of oligomers, what we did was we took a trick from Cliff Brangwine's lab, and we took the Cry2 oligo protein, which is a bacterial rhodopsin that oligomerizes in response to light. So if you just take an M-cherry Cry2 and hit it with light and put it into neurons, hit it with light, you can see granules that form, remove the light, the granules go away, add the light, they form, remove the light, they go away. If you add tau to this, what happens is you see granules that form, remove the light, the granules are still there, but not as much. Add the light, you get more, remove the light, they're even more stable. And this is quantified here, where you can see the increase in granule accumulation when you have tau attached to this Cry2 oligo system. And this video shows you what it's like over 8 minutes, where you can see these kind of granules that form as you shine the light. Now, a lot of reviewers kind of were critical. They said, oh, all that junk on tau, that's not real. That can't be biological. And so we did a lot of work to show, in fact, that you could show the oligomerization and fibrillization through many different methods. We could show it by gel electrophoresis, by native gel electrophoresis. We could use antibodies to detect it, multiple different ways. And when we started looking at the biology of the neurons in response to this, it was exactly like the biology of the neurons when we just put in tau and gave a stress. So we could see the accumulation of stress granule pathology of TO1 in the cytoplasm as we shined light on these neurons. And we also showed that as you shined more light on the neurons for prolonged times, you could get death. Now, I want to tell you at the outset, this is not a lot of light. I quantified the amount of light for going through the microscope with a Thor optical quantifier, and it's actually, you don't need a laser. You can just have a regular LED. It's like the amount of light from your pen light shining on it. And I have a nice picture of Lulu standing next to the light, shining next to her. And so the neurons, normal neurons exposed to this amount of light, are completely fine over either 3 cycles of exposure or 60 minutes of light. But if you have the tau Cry2 oligo construct, you can see that you have fewer cells, you have more LDH, and you have caspase-3 activity occurring, showing that you can get that, in fact, with time, you also get toxicity. Given that it's not a lot of light, that enabled a larger scale application of this. Because now we realize what we could do is take an entire dish and just forget about that laser or anything, just take an LED light, shine it on the darn dish, induce oligomerization, and then immunoprecipitate the tau, do mass spec, and see what's there. And so what we did is we immunoprecipitated it. We went and compared it to either dishes that hadn't been exposed to light or dishes that had been exposed to light but didn't have any added tau to it, and dishes that had tau, the Cry2 tau, exposed to light for either 20 or 60 minutes. And then we did mass spec. And when we did mass spec, we saw a lot of interesting things. First of all, we saw a lot of things that perhaps we would expect to see, like proteins already linked to Alzheimer's disease, like PCOM or clusterin, or ApoE. And then in addition, we saw a number of RNA binding proteins, including at the very top, HNRMP A2B1, which I'll come back to in a moment. But another thing which we saw in here, which I will explain and show some pictures of in a little bit, is the lamins. So we saw lamins and lamin binding receptor, as well as many other RNA binding proteins. So this is suggesting that when we are oligomerizing tau, what's happening is it's associating with many of these RNA binding proteins that are associated with stress granules, and also through the lamins with perhaps nuclear membrane proteins. Here's a summary of what we saw. And you can see many of these RNA binding proteins that are 20 minutes or present throughout, or just at 60 minutes. And you can see the RNA splicing being important in there and RNA metabolism. But after 60 minutes, as you start to get toxicity, you could also see some signs of cell death occurring. We validated this, both immunoprecipitating the tau cryo-2 constructs and showing that we could reproduce what was in that mass spec, but also immunoprecipitating tau from P30S mouse or human brains, and showing that in all these situations, we could also show the association of tau with each of these proteins, which suggests that this has nothing to do with the extra cryo-2 stuff on it, but is an innate tendency of tau to associate with these proteins, perhaps in disease under stress. So I mentioned the lamin 2 protein, the lamin A and B proteins, and looking at this, we saw that using the cryo-2 system, we could really see these striking of tau on the nuclear membrane. And for those of you who are come out of the ALS world, this will sound very familiar because C9orf72 dinucleotide repeats have been shown to disrupt the nuclear membrane, TDP has been shown, and so we were interested in looking at what was happening with tau. And sure enough, as we shined light, if you look at lamin B2, you could see that the distribution of lamin B2 spreads out and becomes disrupted and quantified here. And if we went and looked in transgenic mice or in human cases, we saw the same thing. Here's the normal LBR. We also saw the same with lamin B2. But in animals, as disease progressed, you could see the redistribution of the lamin in the nuclear membrane, suggesting that the pathology is inducing dysfunction of the nuclear membrane. Now, the rest of the talk is going to focus on this HNRMP A2B1, which was really interesting because it's an RNA binding protein. Mutations in HNRMP A2B1 are known to cause motor neuron disease, frontotemporal dementia, and myopathies. And it's associated with stress granules. So we went, and it was the top of the volcano plot. So we went and looked in P30S mice, and what we saw was that as the pathology progressed, we saw an association between cytoplasmic HNRMP A2B1 and oligomeric tau in the cytoplasm. So in the young mouse, it's all nuclear. In the older mouse, it's cytoplasmic. We validated this by proximity ligation assays, and this is quantified here. This is also important pragmatically because it turns out TIA1 antibodies are really bad. I've spent so much money trying to make good TIA1 antibodies, and it just has befuddled me each time. But the HNRMP A2B1 antibodies are really good. So it makes it much easier to look at co-localization between tau and RNA binding proteins using HNRMP A2B1 as a marker instead of TIA1. The question is, why is tau associated with HNRMP A2B1? We looked at the literature, and the results suggested some striking hypotheses. First of all, there's a lot of evidence. So HNRMP A2B1 is very abundant in the nucleus. And in the nucleus, it's an indirect reader of a methylation event on RNA. So when RNA is transcribed, it's methylated, and the binding of HNRMP A2B1 controls whether it's kind of degraded or allowed to mature. In stress, a small fraction of this methylated RNA goes out into the cytoplasm, and it accumulates in stress granules. So immediately one kind of has this suggestive link between M6A, HNRMP A2B1, and tau. And we wanted to explore whether this actually was a complex that was forming in disease. So the first thing we did was we looked at the P301S mice and we asked the question, what happens to M6A? And sure enough, we saw that as disease progressed, you could see an accumulation of M6A in the cytoplasm. And it co-localized with corresponding accumulation of HNRMP A2B1, just as the literature suggested it might. And we proved this using proximity ligation assays for M6A and HNRMP A2B1, quantified here. Next, we looked at the relationship between M6A and tau. And again, we found that M6A accumulates in the cytoplasm and co-localizes with tau, honestly, even better than it co-localizes with HNRMP A2B1. We're exploring the function of this. I'll be telling you a little bit about this, but among the things M6A is known to do is to promote, it promotes phase separation of proteins more so than does regular RNA. So it's easy to imagine why it might show up preferentially in stress granules. When we went and looked in humans, the results were quite striking. So we went and looked at human AD cases at different stages, and you can see that in a healthy control, the M6A was mostly nuclear. But as the pathology ensued, one could see more and more accumulation of M6A labeled RNA in the cytoplasm, and essentially a loss of nuclear M6A from those same neurons, and co-localization with HNRMP A2B1 and TOMA2 as shown with these diagrams. The total amount of accumulation was striking. There's roughly a fourfold increase in total M6A as determined by ELISA. And if we immunoprecipitated tau and look for M6A associated with it, we saw a sixfold accumulation of M6A associated with tau. We're still exploring why this is and how this occurs, but I want to convey to you one short, simple experiment that I think is helpful to think about how to approach disease when using cell lines, and it conveys insight into this M6A accumulation. So Jenna Libera in my lab took some U2S cells, nothing to do with Alzheimer's disease, neurobiology, or anything. And she looked at G3BP, M6A, and M6A under basal conditions. G3BP is cytoplasmic, M6A is kind of mixed nuclear cytoplasmic, and they're not co-localized. She then looked in response to acute treatment treatment with 500 micromolar arsenate for 1 hour. This is what the field classically uses to examine stress. And what you could see is these nice RNA granules forming from a G3BP1, nice stress granules, and they co-localize with M6A as the literature suggested they would. So that all looks good. But the thing is, neurodegenerative disease is not an acute stress. It's a chronic stress. And so the question which Jenna was trying to understand was, well, what happens with a chronic stress? So when she took these cells and instead exposed them to a lower dose of arsenate, but for a longer time to kind of mimic what might be a chronic stress for these cells, now she saw a different picture. No longer did she see stress granules. The G3BP was kind of now diffuse again, but the M6A was showing this striking cytoplasmic accumulation, which I would submit to you is somewhat like what we're seeing in the brains of the Alzheimer patients. We're exploring what this means, but it gives us a simple biological system to begin to understand this biology. Another way we've explored understanding the biology is to kind of hack the system. So when you look at M6A, how it gets there, there are basically three things you have to consider. First, what puts the M6A on the RNA? Well, there's this writer complex that contains these enzymes, metal-3, metal-14, WTAP, as well as some other RNA binding proteins that associate with the complex. So these we call writers. Then when it goes out into the cytoplasm, what recognizes the M6A are direct and indirect readers. The classic direct readers are these YTHDF1, 2, and 3 proteins. And YTHDC2 is what actually shuttles the M6A out to the cytoplasm. And then once you have these M6A labeled RNA, sometimes you want to remove it. And so there are these proteins called erasers, FTO and ALKBH5, that that remove this. So we went and first we looked and quantified what happens to these various elements in the brains of P30S mice. We have a general hypothesis that perhaps, much like blood pressure, a short burst of M6A is good, just like a short burst of blood pressure is good when you're exercising, but persistent M6A may be deleterious in the same way where that persistent high blood pressure is deleterious. So when we looked at the P30S mice, what we saw was that metal-3, that's the writer, goes up with disease. On the other hand, ALKBH5, the eraser, goes down. So that provides a very simplistic, simple model for why we're accumulating M6A with disease, because you're getting more ridden, less removed, hence the accumulation. We're in the midst of completing an M6A RNA-seq to understand specifically what genes are changed, but already right off the bat, we can see that a lot of things that are changed are synaptic, kind of things related to synaptic function. So the next thing we could do is say that we could kind of knock down elements of the system and see what happens. So we took metal-3 and knocked it down. Before I show you our results, I want to show you another paper that came up in the literature about the same time as ours, which might seem confusing, but I'd submit to you it all fits into a picture that begins to make sense. So Zhao and colleagues knocked down metal-3 and then in a wild type animal, and they looked and said, what happens? And if they took a wild type animal and they knocked down metal-3, in fact, they saw fewer neurons and they saw more signs of cell death. So that would suggest to you that not so good, knocking down metal-3. And when they looked at cognition, the animals didn't do quite as well. And perhaps that's not surprising because M6A is required at the synapse where it controls a lot of functions related to translation, decay, processing. So, so perhaps under normal basal conditions, you want to maintain M6A. But what if M6A is elevated chronically? Well, to explore that, we took we went back to our Cry2 system. We exposed the neurons, the tau Cry2 neurons, to light. And so you could see that we got a lot of M6A in the cytoplasm. And we then hit them with either siRNA, a control, or siRNA to metal-3. And you can see that the control, we got lots of M6A in the cytoplasm, lots of A2B1 in the cytoplasm. But when we knocked down metal-3 to reduce the amount of M6A, we prevented the translocation of HNRP A2B1 to the cytoplasm, and the neurons actually looked better. We took this in vivo. we did the same thing in looking at HNRMP-A2B1. We did the same thing with HNRMP-A2B1 in primary neurons, got a result that I'm not going to show you, that it was good to do that. And we went and took the P301S mice, injected on one side, control siRNA, on the other side, siRNA for RH&RMP-A2B1. We waited a month and then looked at what happened to tau accumulation and to neurodegeneration. And so what you can see is that if you just take a mouse and you inject it with vehicle, the control and HNRMP-A2B1 don't really do much at all. So we don't have to worry that it's causing trouble on its own. But if we propagated oligomeric tau, remember from earlier in this lecture, I told you that oligomeric tau induces, can induce more tau pathology, can induce cell death and stress response. What you can see is that, as expected, we got lots of tau pathology, and as expected with the control shRNA, we got lots of signs of cell death. But if we knock down HNRMP-A2B1, shown here, we could reduce the amount of tau accumulation and reduce the amount of cleaved caspase-2 quantified here. So this gives rise to the following idea in summary, which is we think what is happening is that in our Cry2 system, the Cry2 is causing tau to dimerize. In a normal biological system, it's phosphorylation that's causing tau to dimerize. Then the dimeric oligomeric tau is recognized by HNRMP-A2B1. We actually did studies of this to explore whether it was the phosphorylation or the oligomerization that was important. And much to my surprise, I found out it was the oligomerization. Phosphorylation can speed up formation of the oligomers, but even, and which would bind HNRMP-A2B1, but even if you allowed enough time, the oligomeric tau would be recognized just as much by HNRMP-A2B1 as the phospho-tau. And then this forms a complex that recognizes M6A RNA. And as a result of that, it then stimulates translational inhibition. We showed that, but for reasons of time, I didn't show you that in this talk. And if this goes on for a long period of time, you start to get toxicity and dysfunction, disruption of the nuclear membrane. So this gives rise to the following hypothesis. M6A under basal conditions, that's good, you want it. But if you have persistent elevated M6A, it promotes tau pathology, which is bad. Also, it causes a persistent translational stress response, which is bad. And that by decreasing M6A, metal-free, or HNRMP-A2B1, that we can reduce the amount of pathology and provide neuroprotection. Taken all together, this gives rise to a model of disease and begins to explain why, when we think back to the genetics of Alzheimer's, we don't see much for tau. I'd like to propose this hypothesis, which is that in order to define something as Alzheimer's disease, you have to accumulate A beta in the extracellular milieu. So anything that leads to the accumulation of A beta will increase the likelihood that you're going to get a diagnosis of Alzheimer's disease. That can be mutations in APP or presenilins. That can be cardiovascular disease, diabetes. The A beta combines with inflammation to cause toxicity, which is mediated by phospho-tau. Other pathways likely mediate TDP-43, but I don't yet understand what those pathways are. But the phospho-tau, it's clear, forms oligomers and stimulates a translational stress response and formation of stress granules and all these proteins. So if you have risk factors that come in at the top and stimulate A beta, you get diagnosed with Alzheimer's disease. If you have risk factors that come in and directly stimulate the accumulation of phospho-tau, such as mutations in tau, then you won't have A beta, so you can't get the diagnosis of Alzheimer's disease. Instead, you get the diagnosis of frontal temporal dementia. If you have mutations or stresses that come and directly stimulate the stress response through the pathway of the RNA binding proteins, then you get accumulation of these RNA binding proteins, P30, and you get what we call ALS. And so these generally fall into the same kind of pathway. It just depends on which point one enters the pathway. So in conclusion, I showed you that tau forms membraneless granules with RNA binding proteins in vivo and in vitro, and these granules contain stress granule proteins that are likely linked to translational regulation. Tau undergoes phase separation, a process that's facilitated by T1, stabilizes tau oligomers. And in fact, in the presence of T1, we start to get tau forming phase separation at concentrations that resemble those seen in a neuron. We showed that O-tau forms a complex with HNRMP A2B1 and M6A. And HNRMP A2B1 appears to be an indirect reader for M6A. The M6A accumulates in the pathology, both in P30S mice and in the human brain, and co-localizes with both O-tau and HNRMP A2B1. This complex, also shown, contributes to the process of neurodegeneration. If we knock down either HNRMP A2B1 or metal-3, it reduces tau pathology and reduces neurodegeneration. And finally, we showed that since the M6A increases in parallel with pathology, we're beginning to explore whether it might be a useful biomarker for disease. I want to acknowledge everyone who's contributed to this. So, the bulk of the work was done by Lulu Zhang, and that's all a lot of the M6A work and the opto-tau. The work on phase separation was done by Peter Ash and an undergraduate, Eric Lay, who's now at Caltech. Jenna Libera did the experiment on the U2OS cell, and everyone else in the lab just makes this entirely possible. And of course, I couldn't do this without funding, which is so important, and also all of my collaborators who kind of help in all aspects of this work, as well as people at BU.